Journal Club, brought to you by Life Extension Advocacy Foundation, and I'm Oliver Medvedik, coming to you from New York City right now at the Cooper Union, as per usual. Before we go any further, I just want to remind people that we have our third annual Ending Age-Related Diseases Conference coming up soon. We're still not in spring quite yet, but we're already getting ready to launch our conference in August, at the end of August, which will be taking place in New York City. Slightly different venue because we've got more and more participants, so it's going to be bigger and better than ever. It'll be at the Icahn School of Medicine at Mount Sinai, which is also in Manhattan, slightly further north uptown. So do check out our Leave website for our upcoming Ending Age-Related Diseases Conference, and we have a ton of great speakers, and we still have a few speaker slots that we're filling in right now, but things are falling together quite well. So, if you haven't gotten tickets yet, I don't know if they're on sale yet, but check out the website. So, letting you all know that's coming up. So if you weren't able to get a ticket last year, 2020 is your year. All right. So what are we doing today? Well, today we're doing another paper from the laboratory of Liwei Sai and collaborators. This is a paper that came out later last year. So it's not this year's paper, it's last year, but it's a really good follow-up paper on the work that they performed earlier, which involves inducing gamma oscillations in the brain and basically a non-pharmacological intervention, so a non-invasive intervention that could potentially ameliorate or reverse dementias and Alzheimer's disease. So it's really cool that there are these out-of-the-box approaches that are being studied by labs that are non-pharmacological, meaning it's not a pill that's being worked on by big pharma, but these approaches that are non-invasive and might have a really huge impact. So I'm really psyched about this. So I just want to give you a little bit of background in case this is all new to you, because it is quite new. So basically, the brain is very complex, and a lot of its neurons have to fire in synchrony, right? So things have to get synchronized from one region to the other, and basically you can use an electroencephalogram to measure, multiple neurons firing it all at once in synchrony, and when they fire, they oscillate, right? So they give off these oscillatory spikes. And scientists have basically given names to these synchronized firings depending on how many hertz they're firing, right? So 1 hertz is, I believe, 1 cycle per second. So you have delta, theta, alpha, beta, gamma frequencies. And gamma, which is from 25 to 140 hertz, according to a review article that I'm reading, is linked to normal visual consciousness and rapid eye movement sleep. So basically when you're aware, right? So these are gamma oscillations. And the reason gamma oscillations are important in the context of Alzheimer's disease is there was a paper published earlier that showed, I get the reference to that paper, people with Alzheimer's disease show a reduction in gamma oscillations, right? And this paper, I believe, was Koenig et al. Neurobiology of Aging from 2005, so about 15 years ago. So, that led to a lot of follow-up research to see if inducing gamma oscillations, if it's not just correlative, but, there's a positive role there, if inducing gamma oscillations could basically have a beneficial impact on dementias. And there are a number of studies done to see if they can be induced in humans, and the answer is yes. So that led to a paper that we covered a while back, which was Liwei Sai's paper, Icarino et al., I believe, Nature from 2016, where they basically used a mouse model. So this 5X FAD mouse, which is basically an Alzheimer's disease mouse. This mouse has 5 different genes that are all mutated, that have different mutations that induce beta amyloid plaque production and Alzheimer's disease phenotype. And they used, in that paper, they used solely visual stimulus. So they used flickering light at 40 hertz, which is within gamma frequencies. So you had these pulsating frequency of light. And that would entrain neurons in the visual cortex of the mouse. So basically neurons coming from the eye to the visual cortex, and those neurons would flicker at the same frequency. And it turned out that in this mouse model, you have clearance of beta amyloid plaques and improved functionality, memory in these mice when subjected to this treatment. So this follow-up paper that came out several years later is looking at a combination of visual and auditory stimuli, also within gamma frequency. So you can imagine, your body is receiving inputs from a variety of different sensory organs, and some of these inputs can be stimulated at 40 hertz quite easily. So visual is one, obviously. Auditory is another, so you can use sound pulses or clicks, which they did in the paper, and probably vibration, tactile as well. So those are probably the three major sensory stimuli that you can induce at a 40 hertz frequency. So I don't think they've done tactile here, but they've done auditory and visual. And the reason that they're switching to auditory is that, and also in this paper, which we'll go into in a minute, they're actually combining both visual and auditory to see if a combined approach of inducing gamma entrainment in both the visual cortex and the auditory cortex at the same time is better than just one stimulus. Short answer is, according to the mouse model here, the answer is yes, it is better if you do both at the same time. And the reason they picked auditory as well, probably, number one, it's easy to do. Why not, right? It's auditory, just add clicking sounds to the experiment. But it turns out that the auditory cortex is also closer to the hippocampus region in the brain than the visual cortex, and the hippocampus is what's responsible for memory consolidation, which is one of the, for humans at least, one of the first areas of the brain that gets impacted is the hippocampal region. So the authors here reason that if we can stimulate the auditory cortex, maybe some of the effects can affect the hippocampal region as well. And the answer is yes, at least in this mouse model. And they go on to do some more experiments where they look at other regions of the brain that are affected that were not seen to be affected, I believe, in the original paper, which just used visual stimulus, which is the prefrontal cortex. So obviously your brain has a whole bunch of different regions, and Alzheimer's disease and dementia's beta amyloid plaques affect eventually different regions of the brain. And one region that's required for higher processing is your neocortex and prefrontal region, which is where you have higher cognition taking place. And it turns out that if you do both auditory and visual stimuli, a combined approach, you start seeing effects, meaning clearance of plaques in these prefrontal areas that you don't see if you just have a visual stimulus happening. So bottom line is, having a combined approach seems to have at least an additive, if not, a synergistic effect, whatever that means in this context, but a better effect, meaning that you have other regions of the brain affected positively that you normally would not have affected. So that is basically the summary of the paper and the background to the paper. So let's go to the paper itself. There's a lot of data here, so I'm going to probably skim over some of the data because a lot of it's basically repetitious in a good way, but basically a repeat of, in some ways, of what they did in the original paper that we covered, which is with 40 hertz light stimulation, but basically seeing that it's repeatable in the context of auditory stimulation. And the answer is yes. So let's take a look at the paper. So here it is, Martorell et al. So this is Liwei Tsai, his final author here, last author. And this diagram here is a nice little summary of the entire paper. Basically a mouse with a speaker here showing the auditory stimuli, and also combined, right, with a little flickering light and a speaker. Just one word of caution, you could do this stuff to yourself quite easily, right? I mean, it's just 40 hertz sound stimuli and light flickering. You could certainly buy devices that are commercially available. You can go online and find YouTube videos of stuff flickering at 40 hertz or any frequency that you so desire, depending on how well your monitor could support that. But just a word of caution that these gamma frequency flickers are in the frequency range of inducing epileptic seizures for certain people. So just keep that in mind. So you might want to buckle yourself into a seat or something if you're going to try it. Never experienced it before. Okay, so all right. So and just another note: these experiments have moved on from mouse trials. There's several labs attempting to replicate this in humans. In phase 1, phase 2 trials, and Li Wei Sai, along with their collaborator, Ed Boyden, who's a neuroscientist at MIT, they formed a company called Cognito Therapeutics in Cambridge. I believe they formed that in like 2017 to test, this system. Okay, so what is this all about? So, well, this stimulus here, paper is multisensory gamma stimulation ameliorates Alzheimer's associated pathology and improves cognition, which is basically what I sort of been saying as an intro. And the protocol has this acronym, which is GENUS, G-E-N-U-S, I believe, GENUS, which stands for Gamma Entrainment Using Sensory Stimuli. One of the S's probably is dropped. The EN is probably first two letters of entrainment. So a little forced, but there it is. Gamma entrainment using sensory stimuli. So the GENUS protocol. And the sensory stimuli could be one or a combination thereof. So, so what are they looking at? So I mentioned that they're looking at several different regions of the mouse brain. They're looking at the auditory cortex, abbreviated AC. So auditory cortex, hippocampal CA1 region, so I mentioned the hippocampus, and then they look at, later in the paper, they look at the prefrontal cortex of the mouse, right? So those are kind of the three main regions that they basically do analyses on these mice. They sacrifice them and they see what's going on. And they also do, behavioral tests to see what's going on. So this is a huge figure here. Figure 1. Basically, this figure here just shows you that, all these dots refer to basically clicks. So 10 seconds, 25 millisecond, long clicks that are auditory stimuli. And then they want to show here that here's the firing rate of neurons, time from pulse. Essentially, they're showing that these neurons, if they have random clicking or no stimulus, these neurons are not firing in a synchronized pattern. But if you have 40 hertz stimulus, they are, right? So they can measure they have a grid of electrodes that they basically put over the mouse brain, the auditory cortex. And they can measure the rate of firing of these neurons. So this is in the auditory cortex. And interestingly, you get this effect, although it seems to decrease in strength, but in the hippocampal region, they mentioned that it's significant. So you can see that 40 hertz stimulation, this is fraction of cells that are firing. So you get this synchronized firing in the hippocampal region, and also to a lesser extent, but still detectable. It's really hard to see here, but the authors can tease apart the firing of the neurons. You'll get actually auditory clicking. You have synchronized firing, to a lesser degree happening in the prefrontal cortex as well. So basically, I just sort of really quickly zoom through these slides here, but this is essentially just showing that in their protocol, they're able to get, synchronized firing of neurons if they, have a tone going at 40 hertz. And the exact length of each tone, that's in the materials and methods. So, and also the amplitude is somewhere in there as well. So the spiking activity works. So that's figure 1. So that's good that the mice can hear it and process it, and it's detectable in those 3 regions of the brain. Next, they go into behavioral analyses. So in figure 2, again, this is still just auditory. There's no visual, there's no light flicker, it's just auditory. They look at recognition and spatial memory task performance. So again, these are 5X FAD mice. There's a ton of data in supplemental. This doesn't seem to affect normal mice, right? So they looked at normal mice, only these 5X FAD mice, and they have a protocol here where they do 40 hertz stimulation for about a week, which they call habituation, and then they do this these two tests, which are I forget the acronyms. Normal object recognition. Oh, novel object recognition. Sorry, and novel object learning, which is basically they put an object in a cage and the mice basically hang around that object and they go to it, right? So if they put a new object in there, you know, will they, or they remove an object, will the mouse, you know, go up to the object and notice that something's amiss, right? So there's a, I believe that's a standard test for detecting mice recognition. So in both of these, location recognition index and object recognition index, so where in the cage the object was put, novel object location, not learning. The mice, and again, the authors state that this is statistically significant. The mice basically, after this treatment, there was a significant increase in the mice basically scoping out the new object in the cage. So this is the auditory stimulus. So keep me in the comments. One thing I'm curious about is whether the audio, so I'm going to take a little pause here because is whether the audio and visual flicker is happening at exactly the same time, or did the experiment with slightly offsets and maybe the processing time that triggered the slightly delayed one versus the other? That's a very deep question, and I was thinking about that too. I would think that they're happening at the exact same time, but I don't know much about mouse neurophysiology to know if there is some sort of an offset between you know, because you have the auditory cortex, visual cortex, and it's a good question, right? Like if you get both visual and auditory happening at the same time, do you need to offset them to get a 40 hertz 40 hertz effects, gamma oscillations happening in a different region, such as the hippocampus or the prefrontal cortex? I don't know. I am curious about that too. So. Very good question. And I don't know if that's in the materials and methods. Don't recall seeing that. But I'm sure, I'm sure if they did it at the same time, that would be buried in there somewhere. Okay. And then last, they did another standard test for the mice, which is called the Morris water maze, which is essentially a submerged maze underwater, and the mice basically have to figure out their way out of this maze. Probably the walls are submerged, and then they're given several trials and see if they can remember how to get through this maze. The blue line, as you can see here, is 40 hertz stimulation, day 1, 2, 3, 4, different trials of the maze, so they're able to successfully navigate it much better. Looks like there's some learning happening with random and no stimulus, but not as much as with 40 hertz. So just with this, basically just with using this genus application, just auditory stimulus, not just visual, you're getting similar effects with mice. So it's good to see, or hear in this case, that you get that you get not only entrainment happening, but you also get a improved behavioral response that corresponds to response corresponding to improved memory from the mice. So improved memory performance. And so that's essentially the section with auditory stimulation that's just dealing with the functionality of the mice, the behavior of the mice. So now they're getting into, well, what's going on inside of the brain of the mice, right? What's going on with actually beta amyloid plaques? One of the causative, supposed causative mechanisms behind the progression of Alzheimer's disease. So right off the bat, I'm going to tell you that they do see a decrease in beta amyloid plaques in different regions of the brain. That correlates with 40 hertz stimulation at the 40 hertz sound stimulation. So that's a good thing. One thing also they mentioned that's in the supplemental, which I'm not going to go into, but they also see a decrease in neurofibrillary tangles, which is one of the other presumed causative factors behind the toxicity to neurons in Alzheimer's disease. And I believe the neurofibrillary tangles happen earlier, and then beta amyloid plaques are picked up later. So which one is much more important? I'm not 100% certain, but it seems to be having an effect on both. But they look at in what's published here, they're looking at beta amyloid plaques. So they do look at another mouse model that looks at neurofibrillary tangles, and they seem to suggest that it's having an effect on those as well, which is good. So okay, so they look at both soluble and insoluble fractions, two different basically variants of this amyloid beta fragment that gets cleaved and accumulates, and it's stained here. So these are amyloid beta deposits in basically cells that are found in the auditory cortex, the hippocampal cortex, CA1. And then they look at, so these are just two different versions of amyloid beta. So staining, hooked is basically nuclei, and merge basically is merging the two. So all these green globs and these red globs are basically amyloid beta, right? So that's found in and what they're doing in this, when they're doing with these bar graphs is basically you're just getting a quantification of the micrographs here. So average plaque number per area that they look at, you have a significant decrease, with 40 hertz. So no stimulation, auditory cortex, no stimulation, CA1, which is the hippocampal region. So a decrease. Average plaque area also decreases. And let's see, A and B are relative soluble levels in 6-month-old mice. So they basically look at just total levels. And so not just so these are micrographs, fluorescence micrographs, and these are just total levels of plaque. So you can see here just by looking at this that you have a significant decrease, right? So NS is not significant. So 40 hertz seems to carry the day here. So this is hippocampal region and also insoluble. So that's good, right? So amyloid beta plaques go down. Always the caveat is that these are done in 5x FAD mice. So I'm going to stop sharing here. So, that's always been the gripe with a lot of people, is that mice don't normally get Alzheimer's. So you've got to make this contrived mouse model and test your system. That's the best you can do, at least with mice. It's still, we still have to determine whether or not how well this will work and translate to humans. I think there was a trial published very recently, but a very small scale trial where they didn't see much of an effect with light stimulation with humans, and they were probably doing a memory or behavioral test, but it could just be that the protocol is just too short, because when they first did this, genus application in the paper that we covered a while back, I think Li Wei Sai's lab saw that there was only a transient effect if they, applied this to mice 24 hours and then tried to see if there was any decrease in beta amyloid plaques. So they increased the protocol where they would use this genus protocol like 1 hour every day for a week. But this is for a mouse. Maybe for a human, maybe you would need it for 3 hours a day, or 4 hours a day for a month, 2 months, 3. I don't know. I mean, it's there's a lot of unknowns here when you're trying to translate this, from mice to humans. I think it's very encouraging, but but the protocol obviously here has been optimized for mice, not for humans. So it might have to be further fine-tuned, no doubt, for humans. Okay, so. But the good thing is that it's a very non-invasive protocol, and you can play with the parameters pretty easily. So, it should be very optimizable, if you're trying to get this to work in humans. Okay, so they do some more molecular biology here. So let me get to the figure legend spread out all over the place. So auditory genus induces glial response in auditory cortex and CA1 and 5x FAD mouse. So we looked at beta amyloid levels, they go down. glial cells. So these are basically cells in the brain that are not neurons, and they do a lot of supporting functions. And they look at astrocytes as well, so different type of glial cell inside the brain. And basically, one of the things that these cells do is that they clear out damage. So they can phagocytose plaques and basically remove damage, right? So if you have better glial cell response, then you have better clearance of damage. So IBA1, I believe, is a marker for, again, these are fluorescence micrographs, marker for glial cells. So they have these, basically this kind of stringy morphology, so these processes, right? And they mentioned in the paper that if these glial cells are activated, their bodies get bigger, their processes get shorter, but more branched out. So that means they're eating stuff up, essentially. They're phagocytosing things. So here we have no stimulus, 40 hertz. And this is basically, they're also, looking to see if you have glial cells that basically overlay with ingested amyloid beta, which is what's in red here, right? So this would be, if a glial cell if it's inside the cell, it's eating it up. If it's on the outside, it really hasn't gotten to it yet, or isn't even eating or ingesting, phagocytosing, and then later destroying the amyloid plaques. So they look at, so what do they look at here? 40 different regions, auditory cortex, CA1, and then they look at, I think, astrocytes, different type of glial cells. So this is a different marker for them, which is S100B, or I think that's correct. And then GFAP is a marker that correlates with activated astrocytes. Gotta scroll down here to make sure. Yes. So S100B positive astrocytes and GFAP positive astrocytes. So looking at these bars here, microglial count. So you have, after 40 hertz stimulation, you have more microglial, per area. Their body diameter increases, both in CA1 hippocampal region and also auditory cortex. Their average processes link decrease, which I mentioned the authors mentioned, was basically means that they're ingesting or actively phagocytosing. Their processes arborization, meaning how many of these branches they have, that should increase. So you have an increase in that. So essentially you have an activation of glial cells, right? You have a similar effect, maybe slightly less, but you have a similar effect that you have more of these astrocytes go up. So essentially, and in this case, activated astrocytes, meaning that they're ingesting damaged beta amyloid, or in this case, astrocytes ingesting some sort of damage. So so amyloid beta levels, go down, and this correlates also with increased activation of glial cells, which are known to clear this type of damage. So that's with purely the auditory response. Next, we're still dealing with just an auditory response, auditory genus. Here they finally look at the vasculature. So when you have basically activated glial cells, you also have to get this damage out of the brain. So this damage has to be transported through the vasculature. I guess the glial cells have to get out as well, and they stain essentially blood vessels. And the blood vessels basically get kind of thicker. You have blood vessel diameter increases, so you have increased blood flow to the brain. So they use lectin as basically a stain for our blood vessels. Host is just basically nuclei, and they look at CA1 and also AC region, so auditory cortex and hippocampal, and they look at the diameter of these blood vessels, and they measure that and quantify it, and they show that the diameter increases as well. So you have basically improved, as a consequence, improved blood flow after this 40 hertz treatment, which took place over the course of, I believe, a week. for mice. And you have this LRP1, which is essentially I believe it's a transporter. I want to say it's a transporter for amyloid, but I think it might be for the, I'm blanking on what the specific transporter function of LRP1 is. I want to say it's beta amyloid, but it might not be beta amyloid. Darn. We'll hold off and say that it's for beta amyloid. I'll scroll through and double check that later. But essentially what you could see here is amyloid beta overlays with the LRP1 transporter, which is found on the endothelial cells of the vasculature. So basically the green and the red should be basically merging. So you should see an LRP1 transporter co-localizing with amyloid beta, and the co-localization increases. So, so that's all good. So finally, what's even more interesting in this paper is that they do a combined approach, and that's figures 6 and 7. So they leave those for the last. So again, they show that you have both light and sound, and this is where Keith was asking a question: firing rate modulation, right? So how do they, how exactly do they have this combination happening, right? Because you have a click happening every like 25 millisecond long click, and so they do like a burst of clicks. So is it during the duration of the light, or is it offset? I'm not 100% sure. So that might be somewhere in the materials and methods exactly, how they combine the two stimuli. So then they measure, neuronal rate firing, and again, you have correlation with the genus stimulus with the auditory cortex, hippocampus, and prefrontal. But next, what they're doing here is they're looking at glial cells, and they're looking at essentially what they're calling a clustering phenotype response by microglia, which would essentially be microglia contacting or touching these amyloid beta plaques, as they've shown before. But here they're also looking not just at the auditory cortex, but of course the visual cortex as well, and the CA1 hippocampal region, but they're also looking at the prefrontal cortex, which I don't think was, either they looked at it and it wasn't affected. I'm not sure if they looked at it for the auditory stimulation. I think they looked at it, but there was not an effect. But in this case, there is. So this is auditory plus visual stimulation versus no stimulation. And this is kind of a zooming in and a reconstruction, like a computer reconstruction, where you could see these glial cells, which are now registered as dots, which are in contact versus not contacting. If you look at here, not contacting these, well, here's one little contact, but you basically have more contact points, right? So you have basically more overlay. So they're doing this 3D reconstruction to try to quantify in these mouse sections in different regions of the brain to show that you have this microglial response. So the answer is yes, you get a higher microglial clustering phenotype when you have this combined approach, not only in the areas that you expected to see, but also further out in the prefrontal cortex. And in the last image here, and one thing which is not here, and I don't know if it's in the supplemental, excuse me while I zoom here, but I didn't see this, is I'm not sure if they actually, I'm looking for a behavioral analysis. That's what I'm looking for. I'm not sure if they actually did a behavioral analysis to show, could be wrong, but I'm not sure if they did a behavioral analysis to actually show that, you would expect that auditory plus visual stimulation would have a more robust memory response, right, with mice than just auditory or just visual. But I'm not sure if they did that. They looked at, basically in this paper, they did a lot more molecular biology and showed that the effects with auditory plus visual stimulation were detectable further out. So not only do you get an enhanced response in your auditory cortex and visual cortex, so this is basically, again, looking at amyloid beta deposits in different regions, and this is your medial prefrontal cortex. And then after stimulation, you see less of these globs, right? And this is basically, this bar graph just quantifies the plaque number and the plaque area, which goes down, as you would expect. But it also goes down in the medial prefrontal cortex, which I believe was not seen with just visual or auditory alone. And of course, total levels also go down. So this is, what are we looking at here? No stim, this is prefrontal cortex. So this is soluble versus unsoluble. So let's see, do the total levels not go down? 40 hertz, E, relative soluble levels of 6 months followed by 1 week combined with genus. I'm not sure why the total levels, oh wait, sorry, that's, yeah, 40 hertz. So the total levels don't go down in medial prefrontal cortex with just 40 hertz, but audio and visual 40 hertz, they do go down, right? So you do, so, and this is a sagittal cross-section of the cortex staining for amyloid beta, auditory visual. So these are just amyloid beta plaques. So essentially you have less of them. So the bottom line here is that combining this approach, you have sort of a kind of a bleed through response to other areas of the brain that are further out from just the auditory cortex and the hippocampus, going into the, the frontal areas of the, the higher processing areas of the brain, the neocortex and the prefrontal cortex, that you don't see with just in auditory or visual stimulation alone. One thing that, could be a great follow-up is to do a behavioral analysis to see if a combined approach also works better. And of course, can we optimize this protocol, right? So they did week-long treatment with mice, but is it better if you do this for the lifetime of a mouse? Because this is such a non-invasive, therapy that it probably wouldn't be too bad for the mouse to do it for the duration of their lives. If they, have these 5X FAD mice, if you did this for 1 hour a day for the duration of their entire lives, or, a big chunk of it, rather than just 1 week or 7 days, right? And then just do a behavioral analysis or a memory test. So that's, I find this to be really cool because it's a really novel approach. And this could be used in conjunction with a lot of other therapies out there. Perhaps there's a lot of other non-pharmacological interventions that could synergize with this or have an additive effect. People have been, there's been small-scale human trials happening for years with, diet, exercise, aromatherapy. I'm pulling stuff out of my memory banks, saunas, right, which would obviously increase blood flow. It was a Finnish study. And of course, now you have, light stimulation, sound stimulation, vibrational, tactile stimulation within the gamma frequency range. And none of this stuff is really being aggressively pursued by pharma because that's not what they do, right? They make drugs. But I think there's a huge area here for human experimentation because this is relatively safe, if you take, certain minimal precautions, and the payoff could be huge here for, basically minimal expenditure. So being able to quantify these results to see whether or not, this is only good for some dementias, right? Because when we say Alzheimer's disease, we have to remember that Alzheimer's disease is only a subset of all dementias. Dementia is basically a term that measures a a behavioral response, right? So it doesn't get to the underlying reasons behind dementia. If you have a vitamin deficiency and you're basically extremely forgetful and suffering symptoms of Alzheimer's disease, it's still a dementia, right? But it's not Alzheimer's disease. So dementia is just basically means you check off all the boxes for for a certain behavioral responses. But the underlying causes could be as different as a vitamin deficiency due to, well, not eating well because, or alcoholism or something like that. Or it could be Lewy body dementia or Alzheimer's disease or any other type of aggressive, illness that causes the formation of beta amyloid plaques and neurofibrillary tangles. It'd be great to see a huge series of experiments that could be done using all these non-invasive interventions and see, do they, can they work against certain dementias or all dementias or a subset of dementias? Should these treatments be started preventively early on? Will they have more of an impact than if they're, or could they also have an impact if they're, applied later in later stages of particular dementias for individuals? And, that's something of a pet project of mine, is to see if we can combine these interventions and then get this data aggregated online, through a variety of tests that perhaps could be analyzed through either a a tablet or a smartphone, a memory-based test, and basically have data sets of tens of thousands, if not hundreds of thousands of people, because there is a huge potential here that's untapped, I think, that isn't being as aggressively pursued as it can. And if you go to the ALZ, I think, .org website, Alzheimer's website right now, the statistics globally and in the US for Alzheimer's disease and dementias are pretty grim. I could just throw out some numbers, but there's like over 5 million people in the US that have Alzheimer's disease, and numbers projected to go to 14 million by 2050. And by 2050, the healthcare costs just for dementias alone is going to top $1 trillion. And dementia's kill is the 6th leading cause of death in the United States. And it kills more people than breast cancer and colon cancer combined, right? But we hear about colon cancer and breast cancer screening, but you don't hear an awful lot about dementia screening. And the numbers of underreported dementias, or basically lack of screening when individuals go to a doctor, is pretty bad too. I think only like 15% of people get screened for it, if that. It's pretty abysmal, which is quite shocking considering how much of a killer it is, and not just through death, but also through quality of life. Those of us who've cared for a loved one and have seen the direct impacts of a dementia, it's something that, depending on who you are, you may or may not wish it on your worst enemy. It is probably the worst way to go, which is basically seeing someone's essential humanity basically get stripped away and lobotomized from them piece by piece on a daily basis. And I could basically say that one thing to say about dementias is that somebody who's suffering from it, unfortunately right now, the worst days eventually just become the best days, right? It just gets worse and worse, and there's no turning back. So with all those grim statistics and numbers, we really should be doing a whole lot more. So I'm really psyched that we have at least this huge group of non-pharmacological interventions that could be pursued. And and I think there should be a two-pronged approach. There should be much more rapid diagnostics, especially diagnostics using our current technologies. Leveraging our cell phones, leveraging tablets, leveraging online databases, and basically getting as many people and caregivers signed up as possible, and seeing if the interventions work as soon as we can. Because, having clinical trials with 40 participants every few years is just not going to cut it. It's especially if you want to look at interventions where you're trying to see additive effects from many different directions. And like I said, I'm very, very psyched that a lot of these interventions that I mentioned here are pretty safe and are non-invasive. So, we have it in our technology to carry out this research in a much more widespread fashion. So another chat. Yeah, Keith Camito. Yeah, I'd be curious to see variations of milliseconds of one and the other and comparing total entrainment. Yeah, so Keith has been doing a deep dive in this paper and wants to see, what's the best way to entrain these neurons. And yeah, I think there's a lot of room here for optimization. And like I said, there was one paper I have not read. I just read the abstract. There was a human trial that unfortunately didn't see any significant effects using, I think, light stimulation therapy. But I think it was, again, a very small trial, very short term. I think they may have used this, light stimulation 1 hour a day for a brief duration. And I'd like to see, this optimized, right? Because maybe for humans, you need to have multiple entrainments happening all at once, and for many hours and for many weeks or many months. It's, I'm just throwing that out there. Unless you do the experiment, you just don't know. And we should have more of these large-scale trials taking place. Okay, so that is the paper, and I think if we do another gamma entrainment paper in the near future, I'd like to see a human trial paper come out, and maybe we'll cover the one that I briefly mentioned here, which didn't have an effect, and maybe in conjunction with one that eventually does have an effect, and see how the protocols vary, right? Because there is room here for optimization. Which gamma frequency did they use for auditory stimulus? Well, it was 40 hertz. So I think the range for gamma is, based on this review paper, is 25 to 140, pretty broad range. I think they used 40 hertz in this paper, and they used, I think the sounds, right? The sounds themselves were on the millisecond range. There are clicks, so 25 milliseconds. And I think for one of the experiments, they may have done 10-second bursts, right? So it's, that's somewhere in the materials and methods, right? So you can obviously, so click going, at 40 hertz, and then then they would measure the frequency of firing, and click going at 40 hertz. So, so yeah, so obviously if you're dealing with sound, the sound itself, has a duration, right? So, and I don't know what the amplitude of that sound was, or the frequency of that sound. So there's a lot of things you can optimize here potentially, right? Is it a high-pitched tone, or is it a low-pitched tone? But those numbers should be somewhere in the materials and methods. Okay. And again, this is for mice. So mice have different hearing. I think they're much more sensitive. I think they're more in the high frequency range. So, the clicking sounds that work for mice might not be the optimal sounds for humans, and that might need to be modulated based on somebody who has a hearing problem as well. So, but that doesn't mean that you can't have a combined approach in humans, because I'm sure tactile vibration would also work, and it'd be interesting to know if a triple combination of, vibration, and maybe put the mouse on a woofer or a subwoofer or bass.
So for humans, lower, is that what you're saying? Is this your sort of way of saying that you're all about that bass?
I'm all about, yeah, that's right. So maybe
no treble.
Only if you're a mouse. Yeah. So
only if you're a mouse. Fair enough.
Yeah. So maybe we should organize an ending dementia's rave. raise money. And we have basically in the background is like 40 hertz, like thumping and flashing taking place.
I'm imagining some kind of dubstep style music now. Oh,
Yeah.
Keith says he's very chatty tonight, isn't he, Oliver? Makes you wonder why, if he's got time to chat, why he isn't working. He said, I absolutely agree with your comments on dementia, Oliver. Would be great to pursue these low-hanging fruit interventions directly, and thus take this opportunity to mention if anyone wants to support us, please visit lifespan.io/help.
Yes.
Oh, Keith, always trying to plug us there.
Of course. And just as a reminder, if you're tuning in to the tail end of this journal club, we do have a conference coming up, and we will be inviting some speakers who will be speaking about Alzheimer's disease, and it will be at Mount Sinai, the Icahn School of Medicine, at I believe August 20th and 21st, which is uptown in Manhattan, I believe 99th Street and Madison Avenue.
Yeah.
So, put that in your calendar, and do check it out. It will be our third annual Ending Age-Related Diseases conference. So thanks to all of you for making the first two a smash. Like we ran out of, couldn't fit any more people in the Rose Auditorium here at Cooper, which maxes out at 200 people. We will be having it at the Stern Auditorium, which has, I believe, double, if not triple the capacity. Very beautiful auditorium at Mount Sinai School of Medicine. So please see us there.
Yeah, and if you're interested in checking out more about that, you can find out more at lifespan.io/conference. I believe that that now works, that shortcut. So that was cool. So yeah, and to circle back to today's topic, hopefully we'll see more, as Oliver said, we'll see more of this low-hanging fruit tested, and you never know, it may pan out well for humans as well as mice. Let's hope so. Alzheimer's is an absolutely horrific disease to deal with. I know, because I watched my nan die from it, and it's just hideous. So it's personal for quite a lot of us, and I think it's probably the worst age-related disease I could probably think of, because not only does it rob you of your health, it robs you of who you are. And it was terrible watching my nan just completely forget who we were and who she was, which is terrible, really. I wouldn't wish it upon my worst enemy.
Yep.
So there we go. And on that cheerful note, hopefully we'll see some progress in the next few years, Oliver, with things like this.
Yeah.
I think it's fair to say there's been more of a divergence of approaches in the last few years, hasn't it? I was reading an article in, I think it maybe was it the Times, New York Times, that they said that the research for Alzheimer's had been dominated by the one prevailing theory, and very senior scientists were actually actively found out to have been blocking other approaches, and that subsequently that blockage has been removed, and now we're seeing a myriad of different sorts of interventions. The whole amyloid hypothesis isn't necessarily correct, so hopefully we'll see a breakthrough in the next few years.
Yeah, I think it's, at one end of the spectrum, the pessimistic end of the spectrum, there is no pharmaceutical intervention right now that I know of, and I think point blank, there is no pharmaceutical intervention that can slow down or reverse the effects of Alzheimer's disease, like none. There is almost none, or none in the pipeline right now. There was one drug, I think, I forget which company, I want to say Pfizer, that said they they looked at their data and they said, well, it might be working in reversing some aspects of Alzheimer's, but I haven't looked at that data and it's a little iffy. So bottom line is pharmaceutical interventions have failed. That's not to say we shouldn't keep trying, but failed miserably. But, you know, I think there's this galaxy of non-pharmacological interventions which have been, I mean, we looked at the data here, at least in mice, and certainly perhaps in humans. You know, now that we have, you know, we should devote maybe like one episode here to just looking at dementias and all kind of aspects of them, maybe also diagnostics, because, you know, the optimistic side of me is that, well, now we've got interventions that may potentially reverse effects of Alzheimer's that, you know, 20 years ago, people would have said that's impossible. And we are now having diagnostics that can diagnose the effectiveness of these therapies at the molecular level, particularly positron emission tomography, which supposedly, you know, see, you know, plaques and visualize plaques and neurofibrillary tangles. I'm not sure. I think both in living brains in people that are still alive, and to see if an intervention, you know, not only will improve memory using a cognitive test, but actually if you can see a plaque clearance. And until recently, you know, the only way you can definitively diagnose if somebody's plaques have gone away is to dissect somebody's brain after they died. So, you know, now
not really ideal, is it?
No.
Yeah.
So now if you know, if the technology becomes much more standard for positron emission tomography, and we incorporate, you know, online memory tests that could be utilized in huge trials looking at perhaps, you know, both pharmacological but also non-pharmacological interventions like what we just discussed here. And I think if we take it seriously, we should make a huge impact. Now, at the very least, we should at least rapidly find out whether or not all of these interventions work or do not work, and then not waste much time in those directions. So even a negative response quickly should tell us where we should be investing our resources. But right now we don't even have such a protocol in place, which you know, I'd love to see happen. And you know, we have the technology. We just need the willpower to basically do it. So from Victor, is the unknown TPD 43? Wait, what? I'm sorry, this is a lot of acronym. Is the unknown undiagnosed TDP 43? What's TDP 43 dementia?
Come on, Victor, explain yourself. Stop using acronyms. You scientists and acronyms.
Oh, TAR DNA binding protein accumulation. Is the unknown undiagnosed TAR DNA binding protein 43 dementia? Ask me. I don't know. Is that a question, Victor?
Speak to us, Victor.
I don't know the answer to this. You know more about. Is this related to viral infections? No.
He's shy today. He's feeling shy today.
I'm being very cryptic. Sound doesn't work. May mask for discussion. Oh, for discussion another time. Yeah, maybe we can discuss another time. The short answer is I don't know. You know, there's a lot of other alternating theories and hypotheses for what could be a trigger for dementias. Obviously,
and they're all different, aren't they? And we're finding out.
Yeah, we haven't even talked about the microbiome. Yeah. You know, both within the gut and also the mouth, right? Dental health, because you have neurons that traverse from and correlations between dental health and gut health, and you know, and potentially an inflammatory response there, maybe triggering some kind of a cascade which causes, you know, beta amyloid, you know, there's a lot of,
and the drainage theory, the drainage, the the cribriform plate that Leucadia
right
is hoping to intervene by putting almost like a stent, isn't it?
Right. So alternate approaches where essentially you have an accumulation of beta amyloid plaque that can't properly be removed because it's not getting into your lymphatic system, so it's accumulating. So that's kind of a gross anatomical potential defect.
But imagine if it was true. I mean, imagine if it was the case that the brain simply is unable to wash itself clean every day. That's the problem.
It could be like the whole bunch of the blind men trying to describe an elephant. It could just be like like cancer, in that cancer is a single word, but you could have hundreds of different cancers, right? You could have 100 or more different causative causes behind fundamentally in cancer you have an out of control replicating cell that metastasizes elsewhere, but how it gets from point A to point B differs from cancer to cancer. Fundamentally, in all of these dementias, you have an accumulation of toxic garbage, right? And it depends on what it is. Beta amyloid neurofibrillary tangles, and why that garbage accumulates could probably be due to a number of different reasons for different individuals. It could be, it could be that you just don't have lymphatic drain. I'm speculating here, but lymphatic drainage for one subset, but for another subset, that's not the problem. The problem could be that you maybe are lacking a receptor on microglia that causes them to scavenge, that garbage. It could be a number of different reasons why you end up, fundamentally having an accumulation of this, these toxins that can't be removed. And so they might, we might need more enhanced diagnoses, more enhanced, diagnostics that can tell us exactly why a particular individual has this, has this response that's not being properly tended to by the body's defense systems. So anyway, that's a lot to discuss here, but in general, perhaps trying all these non-pharmacological interventions and having a diagnostic system that's online where we can basically see the effects in a huge cohort of people all at once could speed up.
We should take a look at a GAIM as well, GAIM, which is something that we covered in X10 recently. Where they have a not bacteriophage, it's a neurophage that targets amyloids, lots of different amyloids, and it targets the actual fold, and that's how it gets rid of multiple amyloids apparently at once. Is it can actually target the actual fold itself, or sorry, the misfold in the amyloid, which intriguingly raises the question, if it can be translated to humans, it could be potentially used for multiple neurodegenerative disorders. So it could target Parkinson's, CJF, Alzheimer's, all sorts, because the commonality is the misfold. So that's something we could have a look at. They've got a few papers, so
okay.
There's a whole raft of things out there that we could look at if people are really interested in delving into that topic.
Yeah. All right. Well, I think we covered all the high points of this paper here, and I think in the future we will revisit this topic as as we're seeing with Viscidin and flavonoids. We're seeing more and more human trials, with drugs that clear senescent cells, senolytics. Love to see more of this research being translated to clinical trials. And I'd love to see more novel approaches in clinical trials where we could see larger cohorts of people being looked at. So hopefully we'll get to that sometime soon. Hopefully in this year.
Well, you never know. We'll keep on keeping on, and we'll let you know out there what's going on. And you can also visit our brand spanking new website at livespan.io. Some people may have noticed we had two websites, we merged them together. So hopefully that's a better all-round experience for everyone. And of course, you can find us on all your favorite social medias like LinkedIn, Facebook, Instagram, and wherever else we are. All you need to do is look for us by typing livespan.io. Oh, we're also on Twitter as well under that. And YouTube as well. Yes, we're just literally everywhere, so I keep forgetting where we are, which is a good problem to have, I guess. So I guess we'll see you all next time around about the same sort of time next month. And thanks to the Lifespan heroes who help us make this show and many other things and support our work. And if you're interested in doing that and supporting us, you can find out more at lifespan.io/hero. And I think that's about it from me, Oliver.
Yep.
I think so. So we'll see you next time, and we'll let you know what we're going to talk about.
All right,
Thanks
everybody.
for joining us, folks.
Thanks, folks.